Hello and welcome to this video. Myself, Dr. Mohammad Zafar Iqbal. Basically, I'm from Government Arts and Science College, Karwar, and today we are going to discuss about the chromosomes. And before we get into the next session, we have to discuss about the basic information about the chromosome. That's why I have given the title to this video is the introduction to the chromosome. And before we get into any subject, we have to study about the historical background. The history of that particular subject becomes very, very important because we have to identify what are the important milestones which have already been covered and what type of work is to be done in future. That's why the historical background is very, very important. And the chromosomes were for the first time observed by Anton Schneider in 1873. And Anton Schneider is one of the very, very famous person who has done a lot of work in case of the roundworms and flatworms. And he is the person who has given the basic information of the cell division. And before 1873, nobody was knowing anything about the cell division. And during that studies on the flatworms and the roundworms, he has given certain details about the cell division, and during that time he has observed that within a particular cell there are nucleus is present, and within the nucleus you can find that there is a diffused thread-like structures are present, and whatever those structures are there, they were beautifully drawn by the Schneider, and he has not given any name for that person. He has only drawn the pictures, and after that you will find that there is another person called as the Waldier. In 1888, Waldier is the person who has coined the term called as the chromosome. And whatever the term we are using today, it was given by the Waldier. And just for some information, I want to tell something about the Waldier. Waldier was one of the very, very famous anatomists, and he has given a lot of contribution not only to the cell biology, he has also given a very, very important contribution to the anatomy also. And if you can remember, that is the neuronal theory. The neuronal theory was proposed by means of the Waldier only. Then you can also might be knowing about the Waldier's tonsillar ring, and this is a lymphoid ring which is present in case of the oropharyngeal region, and whatever that particular ring was there, it was also identified by means of the Waldier himself. It plays a very, very significant role in case of the immunity. Then we might have heard about the Waldier's gland, and whatever these Waldier glands are present in case of the eyelids, and this is also contributed by means of the Waldier. That's why the Waldier was a very, very famous biologist, and he has given a very lot of contribution to the field of biology. Then if you take the contributions of the Schneider and the Waldier, they have only given the structural details of the chromosome. They have only identified the presence of the chromosome, but they have not assigned any particular function to the chromosome. And this particular work was done by means of the Weizmann. In 1887, the Weizmann is the first person who has identified that there is a chromosomal basis for the inheritance. And whatever that particular theory is there, that particular theory is called as the chromosomal theory of inheritance. He is the first person who has identified that there is a relationship between inheritance and the chromosomes. Whatever the chromosomes are there, these chromosomes have a very, very important role in the inheritance. That's why you can find that whatever this particular theory is there, even till today, we know that whatever this particular inheritance is based upon the chromosomes only. The chromosomes play a very, very significant role in case of this thing. Next, we have to address some of the important terms which are used in case of the chromatin biology or the chromosome biology. And the first term is called as the chromatin. And you might have heard about the chromatin. Sometimes in books we hear both the two terms. One is called the chromatin, and second one is called the chromosomes. And now we have to identify the first duty from our side is that we need to understand the difference between chromatin and the chromosome. Why it is important? Because the same structures at one particular point of time are called as the chromatin, and when they reach a particular stage during the cell division, you can find that the same structures are called as the chromosome. That's why if you take the chemical nature of the chromatin and the chemical nature of the chromosome, there is no difference between the chemical nature of the chromatin and the chromosome. It means that they are almost identical to each other. They are very, very similar to each other. There is no difference at all. Then if you take the physical characteristic feature, then you will find that the chromatin is completely different from the chromosome. Whatever the physical features are there, the physical features of the chromatin are completely different from the physical features which are there in case of the chromosome. Then we have to, first we will discuss about the chromatin. and whatever the chromatins are there, the chromatins are present in case of the interphase stage of the cell cycle. And if you have a idea about the cell cycle, you might be knowing that whatever the cells are there, in majority of the time you can find that the cell is present in case of the interphase stage only, and whenever it wants to undergo division, then only it will enter into the mitotic stage. And if you take this particular interface stage, you can find that whatever these interface stages are there, they have a chromatin, and whatever the chromatin is there, if you take the features of the chromatin, how it looks like a beads on a string appearance. We call it as a beads on a string appearance. Why? It is a very long structure. It's a very thin structure. It is completely uncoiled, and it is diffused. It means that whatever that particular chromatin is there, if you take the chromatin, if you stay in the chromatin, and if you want to observe under the chromosome, you can't see anything. You can't see the discrete chromatin actually. There are different discrete structures are there. Even after that also you can find that you may not be able to identify them as a discrete structure. Why? Because they are very long, they are quiet, and because of the uncoiled, because of diffused nature, it is almost impossible for us to identify the chromatin. Then you can find that if you take the chromatin is nothing but it is a condensation of the DNA which has to be accommodated in the nucleus of the cell, and that's why you can find that if you take the length of the DNA, the DNA will be very long actually. That's why it should undergo certain degree of condensation so that it can get accommodated into the nucleus of the cell. And in case of the chromatin, there is a 50x, it means that there is a 50-fold degree of condensation is present in the chromatin, and whatever that particular chromatin is there, it is more than enough to accommodate that particular DNA into the nucleus of the cell. And next very important point associated with the chromatin is that the chromatins are metabolically active. What does it mean? The metabolically active means they have the ability to undergo replication of the DNA molecule. They also have the ability to undergo the transcription. Whatever the transcription is there, it is also possible, as well as if you take the replication of the DNA, that is also. That's why you can find that whatever, because of the possibility of the replication of the DNA as well as the possibility of the transcription, that's why we call it as the chromatins are called as the metabolically active. Then you can find that whatever the chromatin is there, the chromatin is accommodated in the nucleus, and already said that in order to condense that particular DNA into the nucleus, there is a condensation take place, and whatever that particular condensation is there, that is responsible for accommodating that particular chromatin into the nucleus. Then we have to see the chromosomes. What are the chromosomes? And when we are supposed to call the chromatin as the chromosome, that is also very important thing. And whatever the chromosomes are there, the chromosomes are only visible during the metaphase and the anaphase stage of the mitosis. And if you take the interphase stage, whatever the chromatins are there, the chromatins will undergo further degree of condensation, and whatever that particular condensation is there, that condensation is responsible for the conversion of the chromatin into the chromosome. Then you can find that whatever the chromosomes are there, you can observe them as a discrete structures. You can identify them, you can count how many number of chromosomes are there, and you can identify the types of the chromosome, and you can identify the shape of the chromosome, and you can do a lot of different type of the studies. And whatever this particular chromosomes are there, the chromosomes are generally have arms, and these arms are attached to the centromere. There is a heterochromatin structure is called, and what we call it as a centromere, and to this particular centromere the arms are attached. And then you can find out if you take the chromosomes and the chromatin, the chromosomes are highly thick and they are very compact. Whenever you compare the degree of condensation, you will always observe that the chromosomes are much more compact as compared to the chromatin. Then if you take the condensation, and in case of the chromatin, we have already discussed that the condensation is only 50-fold, whereas in case of the chromosomes, you will find that there is a 10,000 times further folding will take place, and that particular higher degree of condensation is responsible for the conversion of the chromatin into the chromosome. That's why you can find that in case of the chromosome, you can find that the chromosomes, that is the chromatin, will reach the highest degree of condensation. Further condensation is not possible. Then we call them as a chromosomes actually. Then another very important function is that in case of the heterochromatin, we discussed that they are metabolically active, whereas in case of the chromosomes, we can find that whatever the chromosomes are there, the chromosomes are metabolically inert. They neither can undergo replication of the DNA molecule, nor they can undergo the transcription. That's why I can find that no possibility of the replication, and there is no possibility of the transcription. That's why I can find that whatever the metabolically the chromosomes are considered as the inert structures. Then what is the function of the chromosome. Then we have to see the what exactly why the chromatin gets converted into the chromosome. The most important function of the chromosome is that these chromosomes are the structures which help in the segregation of the genetic material into the daughter cells. Whatever you know that during the cell division, a single cell gets divided into two daughter cells, and whatever the genetic material is there, this genetic material has to be equally divided into that is the two cells actually. And in order to have the proper segregation, that is the say we equal half of the chromosome should enter into the one cell and half of the chromosome should enter into the second cell, and whatever this particular segregation of the segregation of the genetic material is nothing but it is the very important function which can be assigned to the chromosome. Then we will see the key features. If you want to identify the chromosome within the species, then we have to see certain key features or the characteristic feature, and whatever these features are there, by using these particular features we can identify the chromosomes within a species. The first point is the number of the chromosome. First we have to see how many number of chromosomes are there. Take for example, if you are studying a particular species, then it will have a fixed number of chromosomes will be there, that will become the number of the chromosome. Then we have to see the size and shape of the chromosome, and what is the size of the each and every chromosome which is present in that particular. Take for example, if there are 10 different types of the chromosomes are there, then what is the size of the first chromosome, what is the size of the second chromosome. In this way, in what way all the different type of the chromosomes will differ from each other. Then we have to see what is the shape of the chromosome. The shape is not uniform. If you take 10 different chromosomes, all of them will not have a same size or the shape. They may have a different size and they may also have a different shape also. That's why we have to identify what is the size of the chromosome and what is the shape of the chromosome. As already said that in the metaphase stage we can easily identify, we can study the chromosome, we can identify the different regions of the chromosome. That's we can find out whatever these particular studies are there, these studies are possible in case of the chromosome. Then we have to see what type of the chromosomes are present. Based upon the type, based on certain parameters, we have the ability to classify the chromosome into some types actually, and whatever those types are there, they are called the types of the chromosome. That is the classification of the chromosome. How many number of chromosomes belong to metacentric chromosome? How many number of chromosomes belong to submetacentric chromosome? How many number of chromosomes belong to telocentric chromosome? And how many number of chromosomes they belong to the acentric chromosome? Whatever this particular classification, this is also very important for the identification of the chromosome within the species. Then we have to see the chromosome banding. What is chromosome banding? By using certain staining techniques, we can produce certain discrete bands on the chromosomes of the chromosomes actually. And whatever this particular bands are there, these are called the chromosome banding. There are different types of banding techniques are available. The commonly used type of the bandings are generally called as the G banding is there. Then we have got a C banding. Then we have got a Q banding, and then we also have got a R banding. And whatever this particular banding techniques are there, these binding techniques are helpful in the micro identification of the chromosomes. If you find any difficulty in the size and shape of the chromosome, then you can go to the chromosome banding, and by using the pattern of the banding of the chromosome, we can identify whether the chromosomes are in what way these two chromosomes are different from each other. That's why you can find that the chromosome banding techniques also very important technique which is used in case of the cytogenetics. Then you can find out the localization of the NORs. What are NORs? The NORs are called as the nucleolus organizing regions, and whatever the NORs are there, this is the region where exactly you will find that the nucleolus organization regions will be there, and that is the region where exactly you will find that there is a distribution of the major rRNA genes will be there. And that's why even if you take this particular localization NOR is also very important. Why? Because if you take this particular NORs, you can find that lot of polymorphism can be identified in case of the given species. Depending upon the what type of the geography is, depending upon the different geographical races, one particular species is belonging to one particular geographical race can have a one type of the localization of the NOR, and the same species living in some other geographical location may have a different type of the NOR. That's why the localization of the NOR also very important for the study of the chromosome. Then we have to, if you take the position of the centromere, it's a very important, because we already said that the types of the chromosomes can only be identified based upon the position of the centromere. Depending upon the position of the centromere, we can classify the chromosomes into 4 types, that is the metacentric chromosome, submetacentric chromosome, telocentric chromosome, and the centric chromosome. So these are the 4 different types of the chromosomes which are there, and that is done by means of the position of the centromere. Next important thing is the how can we identify the chromosomes among the species actually. Then here also we have got certain key features. That is, first we have to identify the number of the chromosomes. Take for example, in case of the human beings, humans have got a diploid number of chromosome is equal to 46, whereas in case of the that is the fruit fly, that is the Drosophila melanogaster, it has got a diploid number is equal to 8. And in this you can find that a particular species will have a particular set of the chromosomes will be there. That's why the number of chromosomes among the species will be different from each other. Then we have to see the size and shape of the chromosome. As we already discussed in the earlier session slide, that is the we can measure the size of the chromosome, we can identify the shape of the chromosome, and based on those these two pieces of information we can give certain important type of the identification criteria to the particular chromosome actually. Then we can also use the types of the chromosomes. So how many type of the chromosomes belong to the metacentric chromosome, how many number of chromosomes belong to the submetacentric chromosome, and how many number of chromosomes are telocentric chromosome, and how many number of chromosomes are acentric chromosome. This can also be useful for the identification of the chromosome among the species. Then we come across another very important thing, that is the sets of chromosome. Then what are the sets of chromosome? Here we come across two important terminology. One is called the haploid set of chromosome, and second is called the diploid set or diploid number of chromosome. And what is the difference between these two? The difference between these two is that there are, in case of the haploid chromosomes, you can find that here you can see that there is a single set of chromosomes will be there, and in case of sexually reproducing organisms, you can find that the gametes will have only one set of chromosomes will be there, and another gamete will have another set of chromosomes will be there. When these two gametes will fuse together, then you can find that one set will come from the father and second set will come from the mother, and that is responsible. Whatever that particular set is there, it is nothing but it is the number of chromosomes which are present in the gamete is called the haploid number of chromosomes. If you take whatever the haploid number of chromosomes are there, it is generally designated by letter called as the small n. N is equal to means they are talking about the haploid number of chromosome. And if you take the example of a human being, humans have got a 23, n is equal to 23, and if you take the diploid number of chromosomes, that humans have got a 46 number of chromosomes will be there. One n of chromosomes will come from the father and second n of chromosomes will come from the mother, and altogether you can find that haploid set of chromosomes means one set of chromosomes come from the father and second set of chromosomes from the mother, that is the haploid. And whatever the haploid number of chromosomes are there, the haploid number of chromosomes are only present in case of the gametes. Apart from the gametes, you don't find the haploid number of chromosomes in anywhere in any of the cells of the body actually. And if you take all the somatic cells, in case of all the somatic cells, you can find that whatever the number of chromosomes are there, always diploid in number, and only in case of the gametes you can find that there is a haploid number of chromosomes will be there. Next term is the diploid number of chromosomes. As already I have told that the diploid number of chromosomes are nothing but they are the two sets of chromosome. One of the set of the chromosomes comes from the father and the second set of chromosomes come from the mother. And 23, if you take the example of a human being, 23 number of chromosomes will come from father and 23 number of chromosomes will come from mother. 23 will become one set of the chromosome and 23 from the mother will become the second set of the chromosome. And when they fuse together during the event of fertilization, that results in the formation of a diploid number of chromosome, and whatever the diploid number of chromosomes are there, it is generally designated by 2n. And in case of all the sexually reproducing organisms, you can find that the gametes fuse together. Fusion of the gametes results in the formation of the diploid, and in case of all the diploid organisms, you can find that all the somatic cells will have a 2n number of chromosomes will be there. Then the chromosome number. If you take the chromosome number, we want to have a general idea about how the chromosomes will be there in case of a different group of animals. First, we'll discuss about the mammals. In case of the mammals, you can find that if you take the diploid number of chromosomes, most of the mammals have a diploid number of chromosomes between 36 to 60. For example, if you take the example of a human being, we have got a 46 number of chromosomes. It means that it is somewhere between 36 to 60. It means that whatever the majority of the mammals are there, whatever the majority of the mammals so far studied, they have the diploid number anywhere between 36 to 60. This is one very, very common thing which is observed. But there are certain exceptions are there. That is called the red Vizcacha rat. In case of this particular visca charity, you will find that there are diploid number. If you take the diploid number of chromosomes, you can find that there is an exceptionally high number of chromosomes are present. Altogether, you can find that if you take the 2n is equal to, it is 102, and that's when you find that it is one of the very, very important exception which is present to the basic understanding. Then another very, very strange and strange example is the Indian Mount Jack. And if you take the Indian Mount Jack, you will find that 6 and 7. Why I have written 6 and 7? Because within a single species, males have a different number of chromosomes and females have a different number of chromosomes. And in case of the females, you can find that there are 6 number of diploid chromosomes are present, whereas in case of the males, you will find that there are 7 diploid chromosomes are there. It means that females will have a 6 number of chromosomes, whereas the males will have a 7 number of chromosomes. Altogether, you can find that within a single species, depending upon the sex of that particular organism, different sex of that male will have a different number of chromosomes and the female will have a different number of the chromosome. Then you can find that whatever the chromosome number, if you want to, these are some of the exceptions, but what we want to conclude is that if you take the diploid number of chromosomes, you can find that there is a species specificity is there. For any given species, you will find that all the individuals belonging to that particular species will have a same number of chromosomes to be there. Take for example, you take the human beings from anywhere in the world, you take the Americans, you take the Indians, or you take the person who lives in case of the Mongolian region. In case of all the human beings, you can find that all of us have got a 46 number of diploid number of chromosomes are there. That's why what we can generalize there that there may be certain exceptions, but what we can see is that the number of chromosomes are generally species specific. And if you take I want to give some brief introduction to the human chromosome. The first studies on the human chromosomes were done by means of the Theophilus Painter in 1923, and he has identified that the 2n number of chromosomes in case of the human is the 48. It is a wrong number actually, and this particular information continued to be there for somewhere about 33 years, and only in 1956 in Tijo, you will find that in 1956 he is the person who has identified the correct number of chromosomes in case of the human being. That is the 2n is equal to 46. That's why you can find that for 33 years the scientific community was under the wrong impression that humans have got a 48 number of chromosomes, but in fact in 1956 it was identified that humans have got a 46 diploid number of chromosomes is equal to 46, and this happens to be the actual correct number of the chromosome. Now we will discuss about the chromosomal variations in case of the vertebrates. First we will discuss about the vertebrates, and once we complete the chromosomal variations in case of the vertebrate, then we will go to the chromosomal variations in case of the invertebrate. And in case of the we will try to take some of the examples which show certain kind of deviations that we have taken the example. And I would like to take the example of a catfish genus called the Corydoras. And in case of this particular Corydoras, you will find that the diploid number of chromosomes are between 44 to 102. Some of these species belonging to the Corydoras have got a 44 number of diploid chromosomes are there, whereas in case of some other species belonging to the same genus have got a 102 number of diploid chromosomes. That's why you can find that you can see that very, very big diversity can be seen in case of the Corydoras. That is the one very, very important type of the chromosomal variations which is observed in case of the vertebrates. Then second example is the South American rodent, that is the Ctenomys. In case of the Ctenomys genus, you will find that somewhere about 30 to 35 different species are there. And if you take all the diploid number of chromosomes in case of this particular animals species, you will find that they lie between 10 to 70. Some of the species of the Ctenomys can have a 10 number of chromosome, diploid number of chromosome, and some others can have a species of the Ctenomys can have a 70. Altogether, you can find that within a single genus with a very, very limited number of species diversity, still we can observe that such a major chromosomal variations are possible in case of the South American rodent. Then you take already we discussed about the Indian Mountain, that is in case of the Indian Mountain, you will find that females have got a 6 number of chromosomes and males have got a 7 number of chromosomes within a single species. Depending upon the sex of the animal, males have a 7 number of chromosomes and females have got a 6 number of chromosomes. And if you take another very, very closely related animal to the Indian Mountain, that is called as the Chinese Mountain. And in case of the Chinese Mountain, you will find that if you take the diploid number of chromosomes, there are 46 number of chromosomes are there. And one more very, very strange thing is that the Indian Mount Jack and Chinese Mount Jack are interbreedable. They can have a viable offsprings actually. Even after such a very, very close similarity, not only in case of their morphological and anatomical structures, even if you take the reproductive compatibility, you will find that they are reproducible, their reproductive compatibility is there. Even after that also can find that in one particular in the Indian Mount Jack, you can find that very, very 6-7 number of chromosomes are present, whereas in case of the Chinese Mount Jack, you can find that 46 number of chromosomes there are there. And this is a very, very interesting study. A lot of work has been carried out. And since this particular discussion is out of context, that's why I would like to not to enter into this particular discussion, and we'll see this particular point in some other video in future. Then in case of the, now we will discuss about the chromosomal variations in case of the vertebrates. And as we already discussed that a huge type of the chromosomal variations are possible in case of the closely related species in case of the vertebrate. Similar type of the variations are also existing in case of the invertebrates. And the first and the notable example is nothing but is the chromosome in case of the family called as, that is the order called the Myrtilidae. And in case of the Myrtilidae, you can find that there are, if you take two genus, one is called the Myrtilus genus and second one is called the Pyrna genus. And if you take all the species which belong to the Myrtilus and all the species which belong to the Pyrna genus, they have a 28 number of chromosomes. That is the diploid number of chromosomes will always be equal to 28. But there is a exception. And what is that exception? The exception is the Pyrna viridis. And in case of this particular, this is the only species which belongs to the Pyrna genus and very, very closely related to the Myrtilus genus. And in case of this particular species, you will find that the diploid number of chromosomes are 30. That is the 2n is equal to 30. That is the very, very important type of the chromosomal variation which is observed in case of the invertebrate. Then we will come across Agrodytes, that is the blue butterflies. In case of the blue butterflies, you can find that Agrodytes byruni. In case of this particular species, you can find that diploid number of chromosomes are 20. Whereas in case of a very, very closely related species, that is the Agrodytes posthumus. In case of this particular species, you can find that 180 diploid number of chromosomes. Now you can see what is the difference in one species of the particular genus. Only 20 number of chromosomes are present, and in another highly closely related species, you can find that 180 diploid chromosomes are there. This is also another very, very important type of the variation. Next example is the Eurasian butterfly, that is the Leptida sinapis. In case of this particular butterflies, you will find that it depends upon the what is the geographical distribution of that species. Same species living in Spain has got a diploid number of chromosomes as 106, and the Spain species, when you take the sampling from the Kazakhstan, then you can find that the diploid number of chromosomes are 56. That's why the same species, same genus, even after that also you can find that these Eurasian butterflies living in case of the Spain have got a 106 diploid number of chromosomes are present, whereas in case of the same species which lives in case of the Kazakhstan, they have got a 56 number of the diploid chromosomes are. This is also another very, very interesting type of the chromosomal variations which is there in case of the invertebrates. And we'll try to, just before we conclude this particular session, we'll try to have a look at some of the common examples. Generally, we have a curiosity. What type of the chromosomes are present in this animal, that animal, that will have made a table. And in the human beings, the diploid number of chromosomes are 46. The fruit flies have got a 8 number of chromosomes. The cat, domestic cat, whatever cats we have in our houses, they have got 38 chromosomes. The dogs, that is the domestic dogs, have got a 78 number of chromosomes. Then honeybees have got a 32 number of chromosomes. The chickens have got a 78 number of chromosome. Horses have a 64 number of chromosome. Houseflies have got a 12 number of chromosome. Cattle have got a 60 number of chromosome, and the tiger has got a 38 number of chromosome. Just before we end this particular video, let us have a recap or the summary of whatever we have discussed in this particular video. We have seen the introduction to the terms. We have discussed about the chromatin. We have discussed about the chromosomes. What is the difference between chromosome and chromatin, that we have discussed. Then we have discussed about the number of chromosomes. We have also discussed about the variations in the chromosome number in the related species. And with this, we are going to end this video. And in the coming videos, I am going to discuss about the rest of the details of the chromosome. Thank you very much.